Um, okay, so at the end of this lecture, my hope is we shall be able to understand the structure and function of the hemoglobin, and then possibly just understand a few types of hemoglobins. And in case you can be able to appreciate the fact that when hemoglobins are abnormal, then you end up with pathology that falls under a group called either hemoglobinopathies or thalassemias, depending on the type and globin that is affected. So without wasting too much time, hemoglobin is a relatively complex structure. And what you see is a diagram of what is potentially found in a red cell with what we call chains or globin chains or polypeptide chains. And there are 4 different types of chains. These chains have different components or structures in them that are resident within the chains. These are what we call the heme groups and ion. Okay, so each of the globin chains or polypeptide chains has an ion and a heme group resident within. Actually, the heme group is a suspended structure within the globin chain. And this suspension occurs as a result of the folding of the globin chain. So basically, we are dealing with a structure that is relatively complex, well knitted in terms of shape, and is resident within the red cell. And you'll also notice that the globin chains appear as either alpha or beta chains. So this is a structure that we want to discuss together, ladies and gentlemen. And my hope and prayer is that by the end of this discussion, we shall be able to understand what a hemoglobin structure is. So I think it suffices for me to say that hemoglobin is a tetramer. And I think you've seen different globins. And its main function is really to bind oxygen and protect that oxygen and deliver it at its intended site. As I did mention, and as you may have seen, they are basically those polypeptide chains that I did mention with alpha or non-alpha globin chains. And I think that was well demonstrated from that structure that I did show. So the main function really of the red cell and hemoglobin for that matter is gas transportation. And this involves transferring oxygen from the lungs to the tissues or removing carbon dioxide from the tissues back to the lungs. So the hemoglobin structure plays this key role of moving gases from either the lungs or to the lungs for exhalation or inhalation. So each red cell, as you may have noted, has about 640 million molecules of hemoglobin. So one single red cell, is highly charged with hemoglobin. And the hemoglobins that we have in circulation would either be hemoglobin A or other types of hemoglobin. But the commonest hemoglobin that we have in circulation either has 2 alphas or 2 betas within its structure. And each of these weighs about 68,000 to 64 or 64,000 Daltons. Other types of hemoglobins that you can find in circulation is what we call fetal hemoglobin or hemoglobin A. All these types of hemoglobins can be found in one single individual, but in different quantities. So an adult might have more than one type of hemoglobin, but in different concentrations. So just like you can see in here, an adult hemoglobin, which is demonstrated as hemoglobin A, has 2 alphas and 2 betas, while hemoglobin B has 2 alpha and 2 delta. Okay, and hemoglobin F, which is fetal hemoglobin, has 2 alpha and 2 gamma subunits. Again, ladies and gentlemen, this is just to let you know that there are different types of hemoglobins that we can have in circulation. Okay, so it should not be distressed at this point on these types of hemoglobins, but just be aware that there are different types of hemoglobins in circulation. And I think in terms of percentages, this is what we all naturally have in our bodies. A normal individual or adult would have almost 96 to 98% of hemoglobin A and 1.5 to 3.2% of hemoglobin A2 and hemoglobin F at a smaller percentage. So basically, depending on what the body is charged to do, any of these hemoglobins can be expressed. You can actually be given drugs to drive your hemoglobin from hemoglobin A and push production for hemoglobin F to higher levels, depending on the disease state that is of interest. So just be aware that our bodies generally have different types of hemoglobin. Having said that then, so what is this hemoglobin? So hemoglobin is basically a hemoprotein. It's a type of protein that is only found in the cytoplasm of red blood cells. You'll not find hemoglobin in any other cell other than in the red blood cells or in the erythrocytes. And it appears in some amounts. A normal adult may have between 13.5 to 17.5 grams per deciliter of hemoglobin, while a female may range between 12 to 15. Now, these ranges vary from one group of population to another, or one type of race to another. So it is not stuck on, or cast on stone. These figures could easily change from one population to another. Having said that, then I think I've already mentioned that each red cell has quite an amount of hemoglobin, and the various types of hemoglobins then would appear in circulation. Okay. Now, for hemoglobin to function, there are proteins within it that must enable the structure to function properly. And these proteins, are stuck together. And the proteins appear in forms of chains. Those chains must be stuck together so that they give the hemoglobin a structure that is concave or biconcave in nature, so that that structure enables the hemoglobin to move comfortably within. The hemoglobin then gives the cell a shape that enables the cell to move comfortably in circulation. Because remember, a red cell should appear as a biconcave cell. That biconcave nature of the red cell is actually a function of the hemoglobin. And so the globins within the hemoglobin must be arranged in a manner and stuck together in a manner that gives the cell the appropriate shape. Okay, and those proteins must then be able to either pick oxygen or release oxygen, because then that becomes the essence of hemoglobin, picking and releasing oxygen at the cellular level. Okay. Now, let me then go straight to what a hemoglobin structure would look like. Remember, I did mention that the hemoglobin is a tetramer. Okay, it is a tetramer. And that tetramer basically is a combination of 4 things. And those 4 things are actually chains. We call them polypeptide chains. Those chains are as a result of amino acids that have been packaged together to form either an alpha chain or a beta chain. Okay, each hemoglobin structure has 4 of those globin chains. It will either have 2 alpha chains or 2 beta chains. Each of the alpha chains is 141 amino acids, and each beta chain, as I did mention, is 146 amino acids. these tetramers appear in a helical format. Okay, so each polypeptide then has this helical structure. Okay, and each helical structure is composed of segments, and the segments are designated as A to H. So there are 8 helical segments in each of the polypeptide chains. Those chains that I did mention when I started off this lecture has segments. And within each of these in each of these structures is ion. Okay, and the ion is suspended within together with the heme component, so that you have heme suspended within together with the ion within the polypeptide chains to form your hemoglobin. Okay, so the question is, how do we synthesize hemoglobin? Okay, so remember, hemoglobin is a combination of 2 things. There is a heme component and a globin component. The globin component is what we have just been talking about as a polypeptide chain or a globin chain. And the hemoglobin, as I did mention, is within the cell. Now, the heme component within the hemoglobin structure is synthesized in the mitochondria of the cell, while the globin is synthesized within the ribosomes. So within the cell, as we all have studied cells, are these structures that can assist in the synthesis of hemoglobin. And so the mitochondria is key in heme synthesis, while the ribosomes assist in globin synthesis. And these 2 processes are well synchronized within the cell. And so heme is basically nothing other than a ring, a protoporphyrin ring, within which is the ion component. Okay, so you have a protoporphyrin ring and ion within it, so that within this ring with ion, when you bring in the globin chain, you end up with hemoglobin. So the heme component is a protoporphyrin ring within which is ion. Okay, and so that heme, as I did mention, is suspended in each of the polypeptide chains. Okay, so each polypeptide chain would have a heme component. All right, each of these, and each hemoglobin has 4 polypeptide chains, as we did mention. It's a tetramer. So each of these tetramers will have 4 heme components. Okay, so heme, as I did mention, is suspended in each of the polypeptide chains. Okay, and therefore the 4 polypeptide chains, which are composed of an alpha chain and a beta chain, another beta and an alpha, would have a total of 4 heme groups. How is heme then synthesized? Remember, we said heme is synthesized in the mitochondria. And this is actually caused or formed from a precursor called succinyl-CoA and glycine. And this is condensed to the delta aminolavulinic acid or DALA. And this DALA through ALA synthetase and pyridoxal phosphate is converted into the heme component. But remember, this will happen within the mitochondria through various oxidative processes. And this diagram basically shows what happens within the mitochondria and some aspects within the cytosol with subsequent entry back into the mitochondria for oxidation to occur for a protoporphyrin ring to be formed, which I did show much earlier. The moment this protoporphyrin ring is formed in the presence of ion, then you end up with a heme component. And basically, this is what happens within the mitochondria or within the cell. So within the mitochondria is glycine and succinyl-CoA, and that leads to formation of DALA. And as I did mention, you have some aspects of synthesis that occurs within the cytosol or within the cytoplasm with the re-entry through oxidation to form the protoporphyrin ring, which in the presence of iron leads to formation of heme. This heme is then released from the mitochondrium to meet the globin chains, which will have been formed from the ribosomes as a result of amino acids being brought together to form either an alpha chain or a beta chain. So the moment you combine the chains together with the heme component, you end up with hemoglobin. Okay, so you notice very quickly that the cell plays a key role either at the ribosomal level or at the mitochondrial level. So what about the globin chains? The globin chains, as I did mention, must combine with the heme component for us to form hemoglobin. Now, there are 8 functional globin chains generally that are formed or arranged in 2 clusters. And these chains are either the beta cluster or the alpha cluster. The beta cluster of globin chains that can be formed is either a gamma delta and a Z globin, an E globin gene. Meanwhile, the A cluster is an alpha and a zeta globin gene. Okay, all these are either found on chromosome number 11 for the beta cluster or chromosome number 16. So these globin genes are actually regulated by these 2 chromosomes. It's either chromosome 11 or chromosome 16 that regulates the production of these globin chains. And any of these globin chains will either form a beta cluster or an alpha cluster. So, a hemoglobin structure will either have 2 betas or 2 alphas at a bare minimum. So, and the various combinations then are a possibility. Having said that, you can then have different globin chains being synthesized, and this synthesis starts as early as the 3rd week of gestation. Now, embryonically speaking, the type of globin or hemoglobin that is formed is called hemoglobin GOA, and it is either GOA1, all right, or GOA2. And these are the various combinations for the hemoglobin that we form. Or you have hemoglobin Portland. Okay, again, you notice the various combinations there that are formed. You can also get, intrauterine, the fetal hemoglobin, okay, or hemoglobin A. Then for the adult, you'll get hemoglobin A or hemoglobin A2. And I think I did mention, and a bit of hemoglobin F. And I think I did mention these combinations previously. So based on the type of hemoglobin that is synthesized, you can have different combinations of hemoglobin based on the stage of development. It's either within the embryonic phase that you'll have different types, fetal or adult phase that you would have different combinations of hemoglobin. So the question is, what or where do these globins come? And as I did mention, you either have chromosome 16 or chromosome 11, leading to either of the 2 globin chains being formed. And as a result of that, then, hemoglobin can be into a tetramer based on the number of globins that are brought together or the type of globins that are brought together. You will either have 2 alphas or 2 beta globin chains to form a hemoglobin tetramer. Okay, and I think this has already been mentioned and I don't need to repeat myself. As a result of that, then, okay, So when you have a chain or rather a sequence of amino acids brought together to either form an alpha chain or a beta chain, then that forms your primary structure. Meanwhile, the secondary structure occurs when you have bonds setting in, so that then your primary structure starts to form or take a helical shape. And that helix is actually supported by bonds. And the bonds could be the hydrogen bonds between various amino acids, okay? When you have various bonds between different amino acids, and actually these bonds are the hydrogen bonds, then you start forming a helix. When you start pushing this helix into a three-dimensional structure by adding more bonds, especially more hydrogen bonds, more ionic bonds, more dipole to dipole interactions, and London dispersion forces, you end up with a tertiary structure, which is a little more sophisticated than the secondary structure. The moment you have this tertiary structure, then basically you are leading towards a much more sophisticated, highly coiled structure that, when brought together, you know, as polypeptides or as chains, can lead to our initial structure of a quaternary structure with 4 different polypeptide chains, you know, so that then you end up with your hemoglobin. And this is what happens at the tertiary level, that you have more bonds setting in. And so you end up with a helix that is a little more complicated in terms of structure, and that is what gives it the tertiary format. And as I did mention, the quaternary structure is basically nothing other than these polypeptide chains being brought together through different bonds. You will have a bond between each of these chains, and those bonds basically bring the 4 polypeptide chains together to form your hemoglobin molecule. Okay, so that is what our hemoglobin looks like. And as we did mention, the ion then plays a key role in the binding, or rather in the delivery of oxygen. Okay, and so ion becomes a key component together with the heme in the presence of the globin chains to assist in mobilizing and moving oxygen within the circulation and to the tissues, or from the tissues. So what are the functions of hemoglobin? So hemoglobin plays various roles. The first role is it acts as a buffer, other than taking oxygen to the tissues. And it does this by binding onto the hydrogen ions, all right, within the red cell. And so it can actually move hydrogen ions and so reduce your pH from acidic possibly to other better states of buffer. The other is it carries oxygen and carbon dioxide, of course, as we did mention, from the lungs and to the lungs. The binding of oxygen to the hemoglobin occurs initially, through various phases, but it is important for me to mention that oxygen binds initially very weakly, but then this increases gradually as more oxygen binds into the hemoglobin. Remember, there are 4 different rings that then enables oxygen to bind in each of the rings. And that binding, causes a change in shape. And we shall be discussing some of the, influencing factors that can enhance either oxygen binding or oxygen release from the hemoglobin. Having said that, then, I want to talk a little more on the states in which the hemoglobin structure can exist. What is important for me to mention at this point, ladies and gentlemen, is that when you take one alpha chain and one beta chain and you combine them together, you end up with what we call a dimer. Okay, I'll repeat that again. When you take one alpha chain and a beta chain and you combine them together to form two bonded polypeptide chains, you end up with what we call a dimer. Okay? So when you combine dimer 1 and dimer 2, you end up with a quaternary structure. I'll repeat that again. When you combine your dimer 1, which is basically also called a subunit, okay? A subunit is also an alpha 1 beta 1 and an alpha 2 beta 2, okay? You end up with a quaternary structure. You'll end up with 4 combined globin chains. Okay? It is important for me to mention that. Now, when you have these dimers, okay, remember within a dimer is an alpha and a beta chain. Between these dimers, okay, are bonds. Between the, within the dimer or between the globin chains are bonds. Between alpha and beta globin chain are bonds. Those bonds tend to be tighter than bonds that combine dimer 1 and dimer 2. Okay? The bonds within alpha and beta as a dimer are called hydrophobic in nature. They are water-fearing in nature. And so it causes a much more tighter bond. Meanwhile, between two dimers, that is, dimer 1 and dimer 2, are relatively weaker bonds. Okay? These bonds are easier to break into. They are called polar bonds. Okay? These two types of interactions are essential when it comes to oxygen binding. I'll repeat again. The bonds between the two dimers, dimer 1 and dimer 2, tend to be weaker. And this then enables us to make it easier for oxygen to combine or enter the hemoglobin structure. Okay, so you have tighter interactions or stronger bonds within the two polypeptide chains or within the dimer, and weaker bonds between the two dimers. Okay, so when oxygen enters the hemoglobin, okay, these bonds, the polar bonds, are weakened. Okay, and oxygen enters the hemoglobin. And so when oxygen enters the hemoglobin, you end up with an oxyhemoglobin. But when oxygen is released from the hemoglobin, you end up with a deoxyhemoglobin. So the bonds here are important in either ensuring an oxygenated form or a deoxygenated form of hemoglobin. Okay. Now, because of the two bonds, the hemoglobin structure can exist in two different forms. It can exist in a form we call the T form or a tense form, okay? Or an R form, which is a relaxed form of hemoglobin. So when you have the bonds with no oxygenation, the hemoglobin is more tense, okay? So a deoxyhemoglobin is actually a tensed form of hemoglobin, while a relaxed form is one where you have oxygen within it. So this hemoglobin here is in the T form, while this is in the R form, okay? And I think this is well elaborated in this diagram where you have a T form or a taut or tense form of hemoglobin because the bonds are much stronger than in a relaxed format when oxygen easily enters the hemoglobin structure. So these are the two states within which a hemoglobin can exist in circulation. It's either in the R form or in the T form, okay? So when oxygen has to enter the red cell, as I did mention, initially it is a lot more difficult for the hemoglobin to enter the hemoglobin. Sorry, it's difficult for the oxygen to enter hemoglobin initially because of those bonds. But the moment you get entry of one molecule or one oxygen into the hemoglobin, then it makes it easier for the next subsequent hemoglobins to enter, and gradually even more easier for the third and the fourth molecules to enter. So the changes in terms of conformation of these bonds is what enables oxygenation to occur. Okay, and as I did mention, because of that, when the hemoglobin reaches a state where you have a lot of oxygen, which may potentially be within the lungs, then it is easier for oxygen to enter the hemoglobin. It will be easier and faster for hemoglobin to enter, sorry, for oxygen to enter the hemoglobin. Sorry for that. And the same could be explained when the hemoglobin is carrying oxygen to the tissues. It is then easier for the same hemoglobin to release the oxygen molecule to the tissues much more than when it is in the lungs. And so this enables hemoglobin to function well when it is carrying either oxygen or carrying carbon dioxide. Okay, so, and I think I've already explained this, that when oxygen concentration in the lung is high, it is easier for oxygen to enter the cell than when it's at the tissue level. Okay, so ladies and gentlemen, I think I've already mentioned that because of this, there are different types of hemoglobins, and the body can be stimulated to give either of these hemoglobins. But naturally, these are the concentrations that you'll find in circulation. Let me talk about then the various types of hemoglobins that we can find within our bodies, or at any one time you could find different types of hemoglobin. If you get hemoglobin that is oxygenated, as we did mention, then that is oxyhemoglobin. If it loses oxygen, then that is deoxyhemoglobin. Okay, when you have the met component entering the hemoglobin, all right, then you have, or the ferric component, then you end up with met hemoglobin. If you have carbon monoxide, okay, the CO2 entering the hemoglobin, such that it poisons that hemoglobin, you end up with carboxyhemoglobin or carbonyl hemoglobin. And this is what causes poisoning generally if someone is restrained in an environment where there is carbon monoxide. Okay, because the carbon monoxide then binds to the hemoglobin and is unable to release the hemoglobin. And so someone gets suffocated, and as a result ends up with injury as a result of the carbon monoxide. Okay, but it can also be bound, okay, non-covalently to carbon dioxide to form carbaminohemoglobin. So when you have carbon, you end up with carbaminohemoglobin. Sometimes hemoglobin gets attached to glucose, and when hemoglobin binds to glucose, you end up with glycohemoglobin or hemoglobin A1C. And this is a hemoglobin that we actually use to check the level at which sugars are controlled in a diabetic. So patients with the diabetes with high glucose levels then would easily enhance or cause glycation of the hemoglobin with glucose. And that glycation causes glycohemoglobin. Okay, and of course we can use this to measure with glucose. If it is well controlled, then you'll have a reduction in the glycated type of hemoglobin. If there is poor glycemic control, then hemoglobin A1C would be elevated. Having said that, ladies and gentlemen, there are factors then that affect oxygen, or rather factors that affect hemoglobin functioning when it comes to oxygenation and carrying of oxygen. It is important for me to mention that when oxygen binds to hemoglobin, you end up with an oxyhemoglobin. But the process of oxygen entering into the hemoglobin can be assessed using an oxygen dissociation or oxyhemoglobin dissociation curve. And we know that oxygen is carried in the hemoglobin, or entry of oxygen into the hemoglobin is a function of the partial pressure of oxygen. So oxygen compared to other parts of the body. And so when hemoglobin is moving in the body and has to lump up this oxygen, there are various factors that can enhance either entry of oxygen into the hemoglobin or reduce entry of oxygen into the hemoglobin. And this can be demonstrated through a sigmoid curve here, where you have oxygenation concentrations occurring within the hemoglobin. So the y-axis is the amount of hemoglobin that has oxygen, while the x-axis is the partial pressure of oxygen wherever it is. Okay, and so if you have this kind of curve, it will give you a sigmoid shape, or a shape that then can assist you to make assessments quickly on how much oxygen can bind onto the hemoglobin depending on the partial pressure of the oxygen. And so hemoglobin F tends to drive this process more towards the left rather than towards the right. And anything on the right means the oxygen concentrations within the cell may be high, and so the ability of the cell to release that oxygen is a lot more difficult. Okay, so if you have hemoglobin S, it's more difficult to oxygenate this cell and more difficult to release the oxygen than when you have hemoglobin F. 2,3-DPG, a high pH will actually make the hemoglobin get easily oxygenated, but then just remember that some of these cells tend to make release of oxygen a lot more difficult. And so as you increase your partial pressure of oxygen, you increase the saturation levels of the hemoglobin. And as I did mention, more 2,3-DPG, a lower pH, and hemoglobin S at a higher partial pressure would increase oxygen levels within the hemoglobin. While on the right side, basically this is just the effect that hemoglobin has on either carbon dioxide and how the acid-base balance is played by hemoglobin. And you can see that the hydrogen ions can be bound by hemoglobin, and this can actually be delivered through the Bohr effect for buffering our acid levels within the system. And so you'll read more on this, and you'll be able to appreciate that hemoglobin does more than just transporting oxygen, and also assists with buffering the pH within the body. And so, as I did mention, you know, there are various substances or pH aspects that can assist in driving the curve either towards the left or towards the right in terms of oxygenation. And this then determines how much either oxygen enters the hemoglobin or is released from the hemoglobin. I want to leave my presentation at that, ladies and gentlemen, and wait for either your comments, and I should be able to share the slides with you for the team to then go through them as you read more on the hemoglobin structure.